Hello, everybody. This is the last week of material for Biology 213. There's kind of a lot going on today. So just a reminder, we need to finish up the plant responses lab. That should only take about 30 minutes. You're gonna look at the experiments that you set up last week. Record what you see and finish any questions that you have in the packet. We'll go over some of that information just as a little reminder, but then we're going to go into the Angiosperm Reproduction lab. And so you're going to be turning both of these packets in today, along with your stomata handout and the full lab report that we gave to you 2 weeks ago. So a lot due today. Don't forget it. Since we have the plant practical immediately after this and we don't have a week for studying and returning material, your TAs will try to get as much or your packets back to you as soon as we can, but I would take photos of pages that you've or on the pages that you've turned in, just so you have that information for studying, or take some notes on the labs. And I also highly recommend going over the study guide that will be posted on BBLearn, and yeah, reading all the backgrounds on the labs and stuff. Let me or your TAs know if you need any, or if you have any questions or anything like that. It will be set up very similarly to the animal practical. All right, so just a little brief, plant responses, little wrap up. So you're going to observe your corn and bean experiments that you set up. You're going to finish your packet and make sure that you have read the directions and answered the questions in detail. I have a little photo down here of just a picture of some gravitropism in roots, and what happens when you remove the root cap. So remember that the root cap, one of its functions is to sense gravity, and so they have these starch granules in the root cap that will help the plant reorient itself to the position that it's in. And if you remove the root cap, theoretically the plant will not be able to respond to gravity. And so that's what you can see in this picture, is that I believe these are corn seeds. So a regular corn seed would put its root out, put its root out, and then it would immediately turn down towards gravity. So you can see that the corn is positioned in different directions. And so if the place where the corn root will come out, which is the pointy side, is pointed down, the root will just grow down. And similarly, if the corn root is placed with that, where the root comes out facing up, the root will come out and then curve immediately down. So you'll take a look at your corn beans and stuff. Just a little reminder about phototropism. So this was the last activity that you looked at, and you should have looked at a plant by a window that looked like this first picture. And so just a reminder that this is a growth in response to light. And so auxin is responsible for gravitropism in gravitropism, but it's also important and controls phototropism. And so auxin is these little pink dots here, and the auxin will always accumulate on the dark side of the plant, and that will promote cellular elongation on that side, on the dark side, that way that the stem will curve towards the light. So this is what we're looking for in the packet. So make sure that you understand this and relay that in your packet. And then I think I have a gravitropism slide, same slide I had last week, but just remember here that we're looking at gravitropism in roots and in the shoot system. With phototropism, we only looked at shoots. And so there's differences in the shoots and the roots in gravitropism. And so shoots are negatively geotropic, so they're always going to try to grow away from gravity, whereas roots always try to grow with gravity. And just remind yourself about where the auxin is accumulating, what side is being elongated, and where it is bending to. All right, I won't spend any more time on that. So after all of that, we're going to go into angiosperm reproduction, which is probably one of my favorite labs, especially because it's springtime out and there's a whole bunch of flowers around campus for us to look at. So the objectives that we'll have in lab, sorry, I'm just moving my little face. Our objectives in the angiosperm reproduction lab is to study the structure of a typical flower. A typical flower in this case is one that is recognized by people that are not plant orientated. And so we have tulips or lilies for you to look at. And we actually this year have 3 types of flowers. So I'll just show you them. We have our tulip, so you can take a tulip for your first flower. We have these big lilies that look like the picture that you have in the background. And then we have these little small flowers. So you can take any 3 of these. The structures are similar, but not completely the same. So you will look at the structure. Oops. Okay, so you'll look at the structure of the tulip or lily, and then 2 other flowers of your choice. And so Dave and I and your TAs will be collecting flowers around campus this week for you all to look at. And as you study the structures of these flowers, you will be looking at the male and female reproductive organs needed for sexual reproduction in flowering plants. And then you'll also dive a little bit more in depth and just look at the pollen grains and observe a dissected pistil, which is where the ovules are. So for lab, I highly recommend reading over the chapter in your book that talks about angiosperm reproduction. I'm hesitant to say a chapter. If you have the 5th edition of the book, it will be chapters 41.2 and 41.3. I completely didn't realize that we had other people or another edition out. So just find the part where it talks about angiosperm reproduction and read a little bit about that. You'll want to know these basic structures and what their functions are. And so the information should be in your book. It's also in the packet. So lots of places to find the information there. So look over that information, and then just in detail, the last thing or what we're gonna we're asking you to do during this lab is for each flower observed, you're gonna follow the dissection procedure in the handout that we've given you. And you're going to fill in the observation sheets. And so we have an observation sheet for each flower that you'll look at. And I just wanted to point out one thing. And so this, oh my goodness, this little text script or this little text photo is from your dissection procedure. And so it's the dissection procedure for the sepals. And on your observation sheet, under observations, we have listed A and B, A and B. And so these are actually, we're asking you questions or specific questions to answer. And so for the sepals, you will answer for A and B, the 2 things that are listed under sepals. So how many sepals does your flower have, and describe the appearance of the sepals. And then in the dissection procedure, you'll also It'll also tell you to remove the flower parts and tape them into position onto a piece of paper. That piece of paper, you only need to tape the first flower onto the separate paper. And so this is what it will look like. And so you can see I got my sepals and my petals, and then my reproductive structures and drawings. And so this will be in lab for you to reference if you need. And again, just to reiterate, you only need to do that for the first flower that you do. The other flowers that we'll have in lab are a bit smaller and a bit more complicated to do dissections and and such on. So if it's too hard to find like how many ovules are in the ovary, don't worry about that. You don't need to know. Or I think it asks you how many ovules are in the locule as well. So don't worry about the super small things, but you should be able to tell on the extra flowers that we bring in, like how many sepals does it have, petals, stamens. You'll be able to see basically all the reproductive structures for both male and female, other than like the female ovule, locule, ovule stuff. For the pollen, it'll ask you to make a wet mount of the pollen. So you can basically just take the anther of one of your flowers and tap it on the slide, the microscope slide, and just that will basically, if the anthers are open or dehisced, the pollen will just fall off, and then you can add some water in a cover slip and observe it under the microscope. So yeah, I think that is all we have. I will get the plant practical study guide up on BB Learn.